hopefully what I'm going to be able to do today for you is to just give you a different perspective on biological farming. A lot of people have the misconception that it's all about organics and that you have to stop using fertilisers altogether and you have to stop using pesticides and herbicides. Obviously we'd like to achieve a sustainable state, but it's more of a longer term process, and we'll be looking at a whole range of different issues in terms of soil health, and the biological side of it is one aspect that I think is really important, that we've got a lot to learn about there. So I don't want to stand up here and preach too much. I'd rather just present some issues and have people discuss it, so try and involve you as much as I can. Hopefully I can provide some new ways of thinking about things, but just to put in perspective, biological farming isn't really a new process. It's been going on for obviously hundreds of years, but obviously we have new technologies available to us today that can help us manage it and monitor it better. So things like microscopes and cameras and different technologies that we can look at our soil, and soil testing methods as well, that is going to open up for the future. And hopefully in 10 to 20 years time I'll be standing here and a lot of the things will have changed, because that's the way technology is going. We've got to keep adapting all the time and keep new information coming onto the front. So some of the other things we want to talk about is to get some practical tips. So obviously we're not doing any outside stuff today, so I'm going to have to talk about it on the board, but hopefully you can relate the information I'm talking about here to your practical situation, to your farm. So hopefully we're going to learn from some others' experiences. Bill Twigg, I've read Bill's little bio about his farm, and hopefully some of the stuff that I'm talking about will tie into what Bill's got to say as well. And challenging your thinking, I think that's the biggest part of biological farming, that we're going into this, or we've been changing for probably the last 20 years, but going through a new way of thinking. I'm reading a book at the moment by Charles Walters, who's the late Charles Walters, he's not alive anymore, but the editor of Acres magazine from the US. And he was talking about, back in the 1970s, a university guy that was on the radio that was talking about burning wheat stubble at the time, and that they knew that it wasn't probably the best way to deal with the stubble, but they hadn't sort of connected the dots yet. They were sort of talking about the next year's crop and the amount of carbon that was stored there and the amount of nitrogen that they were going to need for the next year's crop to actually help start that composting, in situ composting breakdown process. And this university guy said, well, I know it's bad, but what else are we going to do with it? The moral of that story is obviously, if we know something is not working, then we've got to work out other ways of doing it. There are other ways of doing things. We don't just have to go to the main, the most tried and true route. We can adapt things and experiment all the time, and hopefully that's what we'll be doing, over the next 10 years or more in farming. So one of the crucial things that I deal with a lot with my clients is where do they start? They come to me and they say, oh, I want to go down the biological farming route. Where do I start? Well, you've got to start somewhere. So you've got to have, firstly to identify the problem. So what are some of the ways that we could do that? Yeah, so soil testing is the answer there. Obviously, when you're around the farm, you'll have your own indications as well about what's growing, what's not growing. You'll be able to have your paddock histories and all that sort of stuff, and you'll be able to put that together with soil testing information. But for me, the most helpful indicators are soil testing, and not just nutrient testing as well, but biological testing. So a lot of the time I deal with clients who just say, oh, well, I'm just going to put out this, I'm just going to put out, whatever. And I say, well, why? Why are you going to do that? Why are you putting out urea? Why are you putting out sulphate of ammonia? Why do you need to do that? And they say, oh, because, for crop requirements, of course. And you're like, well, have you actually checked what you've actually got there first? Have you actually checked off what you're starting with? And a lot of the time, the two soil tests are linked. If you get a nutrient test back, and most of you have probably never even done any biological testing. Anyone done biological testing before? No? Send a sample off and have a look what's in there. When you get that sample back and you put that together with the nutrient test, it builds an overall picture of how you're actually arrived at those nutrient levels. So if you got virtually no fungi in your soil sample in terms biologically, you can almost certainly see the link between calcium and phosphorus solubilisation there, and also structure and humus production and carbon levels and all that sort of stuff. So it is really valuable that you sort of start thinking about those options. They're a little bit more costly than just your general nutrient test. But if you've been doing nutrient testing for 10 years or so, and you've got a good bank of data there, and you know what your property's doing, and you've never done a biological test, well then I'd really encourage you to do that, because it's a new piece of information that you can use in your suite of tools to work out what you're going to do on your farm. And it directly influences what I do in terms of consulting with people.
So what is the cost?
Yeah. The cost, a basic standard assay is $250 for one sample. I think you can get discounts for multiple samples, but I don't charge any more on what the laboratories charge. So you can get that done. You don't have to go through me. You can do it yourself through a laboratory. But obviously I provide consulting with that to help you work out where you're going with that. But so ultimately, where would we like to be at the end of today, or where would we like to be in the future of our farming? One of the really interesting things for me in farming, and with my partner, we farm just out Canary Island way this way, and so for me to want to be involved in farming in the future, I don't want to be doing it the way we're currently doing it now. I want to be doing it sustainably, and to go down that route, we need to. What is this doing? We need to be looking at triple bottom line sustainability. So not just economic sustainability in terms of it's financially viable, but also whether it's environmentally viable and also socially viable. A lot of people forget the social side of it, but especially for women on the farm, you want to be able to feel that you can have something to do on the farm. And I find that women are often best placed to sort of manage the biological side of it, because they have the time and the interest to sit down and look at the detail and actually monitor it over the time, and the guys tend to be better at actually putting the stuff out and mixing up brews and things like that. So it can be a team focus there. So hopefully by the end of the process, and I guess there's no end really, it's just a journey, but throughout the process we'd like to limit the need for fertiliser use, obviously, because there's a huge input cost that if we can minimise that, we're going to be better off financially. And also limit the need for pesticide use. How would we do that? Obviously the biological organisms that are there play a huge role. So we let the soil biology do it, basically. And also to limit the need for herbicides. And he probably is sitting there thinking, well, how's the biology going to limit the need for herbicides? But as we know from studies, especially ones done overseas and brought to Australia over here with the Soil Food Web, is looking at the biological processes associated with fungal dominance and bacterial dominance in terms of the different forms of nitrogen that are supplied to the plant. Now, what I'm finding is that with higher bacterial dominance, we're getting more nitrogen production, and we're getting higher selectivity for weeds. So we're getting more weed growth. We're actually selecting more for that over time, and we end up with a production system that's not actually selecting for crop production, or if we're not in crops, if we're in perennial systems, horticulture or agroforestry or anything like that, we need to be shifting more towards the fungal side of it. So that's what we're talking about there with the limited need for herbicides. And I guess the other side of that is that we do need to consider the diversity. Now, most of the time in our cropping situations, we're trying to limit diversity. We have been trying to limit diversity for the last 200 years, I guess, in farming. And so every time we take something out of the scenario, we need to be thinking about, well, what is nature going to be putting back into that scenario? If we take something out and we leave a gap, what's going to happen?
Something worse takes its place.
That's usually the case, yeah. If we're not supplying the biology back in after we take it out, we're going to leave ourselves open for pest and disease infection, because there's no cover, there's no predators. And the same in a cropping situation. If we take weeds out and we don't put anything back in, then we're going to have a bare canvas again for nature to come back and put some more plants in there. And usually the things that establish the best in a disturbed scenario is weeds. So we're just selecting for the same scenario over and over again. The other thing about the plant diversity and biological diversity, the link there is the more variety that we can have in terms of our plant growth, the more variety that we can have in terms of our biological health. I don't know how many of you know this, but certain soil organisms are attracted to certain types of plants based on the sugary exudates that are exuded out of the root zone. So if we have just all the one same type of plant, imagine all of you here are the exact same person and you all eat the exact same foods and you all give off the exact same smell and taste and product, then only one thing is going to be attracted to you. So it's limiting the diversity basically. If we can increase that plant diversity, increase the biological diversity, we're going to be much better equipped to deal with pest and disease infestation and also weed growth. The next step to go down that pathway would be, obviously we've come a long way with science and technology and we don't, our understanding of soils has come a long way since people used to think that plants actually ate dirt. We all know now that plants don't eat dirt and they don't just have a big straw-like mechanism on their root where they suck up nutrients. They do take nutrients out in different forms and biology is obviously a major part of that. I guess the main part about the biological side of farming is the uniqueness of plants. We can't forget that plants make their own food. So they've been doing it for, 130 million years or so since the first flowering plants came onto the planet and started evolving. They've evolved to make their own food and to make their own proteins from amino acids. They make their own fats and other proteins and bioflavonoids and alkaloids and things like that that are designed uniquely for that plant to attract different biology and to work with different biology. So everything in the plant has to come from somewhere. If we start to think down that pathway, then we start to think, well, if it's not coming out of the fertiliser, where else is it coming from? On our farm, we've been minimising the fertiliser input, or even in some paddocks not using anything. So if we can grow a healthy crop out of that, well, where's it coming from? It's not coming from the fertiliser. Obviously, the main component that we would be aware of is obviously sunlight, energy, photosynthesis. The plant is making its own food. It also does take up nutrients through the root, but usually in solution. It's not just actually, picking up little particles and taking it up there. And the biology acts as a conduit for that as well. So when we've got things like mycorrhizal fungi happening around the root zone, we're actually extending that ability for the plant to take up nutrients through that solution and through that conduit of the biology associated with the plant root. The other aspect of it too is obviously through the leaves, through gaseous exchange, and we'd be aware of that through some of the foliar sprays and things that we do. And obviously the biggest perspective in my mind is the biological side of it. So taking the benefits of mycorrhizal fungi, bacteria, protozoa and nematodes, and you probably freak out when I say nematodes, I'm not talking about the bad ones, I'm talking about the good ones. So we need to keep in mind all the little critters that are happening there, what they're doing, what functions they're having there and what nutrients they're providing us in what forms and how they're going to feed our plant in order to be able to maintain that sustainability. So obviously between all these different forms by which the plant can uptake nutrients and basically produce proteins to be able to produce grain or produce flowers or whatever it is that we're producing, the soil microbes have to join the dots. And not just to be thinking about it in terms of just the soil surface or underneath the soil, we're also talking about on the outside of the plant, so the plant leaf, the stem, all that soil interface that's between the ground level and the actual crop production canopy. So if we're going to think about bringing back the biology, we've spent a long time, as I was saying before, limiting the diversity. We also spent a long time getting rid of the biology. We've been selecting, for minimum diversity, and we've been minimising potential fungal sites on our root zone by using fungicides and tillage and etc. We're now starting to come back, back to the biological side of it again. But we have to recognise that a lot of that diversity has gone, and it's up to us to be able to put it back again. I guess that's, if we link that back to the soil testing, if we start from a common point and we work out actually what's going on in there, then we can target what organism groups we have to actually put back there. Makes our job a lot easier. So if we're going to foster the biology, one of the things I normally talk about with a lot of my clients is providing a home and a food. If we don't provide a home and the food for the biology, then it's going to be really difficult to keep them there and to keep them interested and keep them working for us. It's basically, they're an untapped workforce, and that, draws into Joel's introduction, untapped paddock potential. If you're not caring about your workforce, your soil organism workforce, then, you're basically missing out. So if we're talking about creating a home for the biology, where would be the critical point that we'd be looking for that? In the soil, hopefully, on the leaf surface. If we think about some of the soils around here, what would be the major, limiting factor for establishing a home for biology in our root zone?
Rainfall.
Yep, rainfall would be one of them, but
Water that transport that nutrient into those plants.
Yep, but in terms of creating a home for the biology aren't living in the soil particle. They're going to live on those moisture and air pockets on the outside of the soil particle, and they're going to exchange those processes there. John? Yeah, humus is a site for the soil biological life. And, where does humus come from? We've got to have carbon sources. We've got to have carbon to feed them, and we've got to have a home to provide them. So if we're going to provide carbon sources with minimum tillage and things like that, we need to think about taking it to the next step. You've got all that carbon out there. Think about what we can do by adding the biology and actually breaking that down instead of having it just sitting there. I know I'm not going to mention the guy's name, but probably fairly well-known farmer from around sort of out that way. No-till for a long time, and he was interviewed, I think, on TV, and he was saying, I've got 5 years worth of stubble here in my paddock, and he was showing, the cameraman, and I just sat there and thought, well, got 5 years worth of stubble there, what are the biology doing? They're meant to be breaking that down. If you've got it still sitting there and it hasn't broken down, what is that telling us? Well, no microbes, or are they not getting the food, or are they not there in the right numbers? Have they got too many predators? Have they got not enough predators? Have we got enough fungi? All those sorts of things start to go through your mind. You start to open up to a different side of way of looking at it, instead of saying, I've got all this carbon here, but I just haven't brought in the microbes and started working with it yet.
What would happen if you put stuff on that pound into the soil to get it working that way?
Well, you can pound it into the soil, you could even sit there with a roller, you could cultivate it, and you could do whatever you like. But if you haven't got the microbes there, then where are they going to come from? They'll either have to be there endemically, in terms of they already have to be there and you provide them the food and the home and they will grow and multiply. But if we're not actually, if we're starting off from behind the 8 ball in terms of we've knocked down the diversity over so many years, and we just start providing the food and we don't worry about providing a home, we don't worry about providing good soil structure and good porosity, because remember I was just talking before about the soil particle, them living on the outside of the soil particle. If we squish up all the porosity and squeeze all the air particles out of it, squeeze all the moisture particles out of it in terms of the space to store moisture, we don't have much soil porosity there. Where are they going to live? It's a multiple effect. We're not just arriving at one conclusion from one issue. There's a host of different factors occurring there in order to get to that stage. Because a lot of the time we can't see what's happening because it's too small for our eyes to see, we need to look at it under the microscope. A lot of the time we're not recognising that we're doing the damage because we can't see the effect. We can only see secondary effects and things that happen perhaps 12 months or 18 months later as a result of something that we might have done before, and by that time we may have done other things that will have their repercussions down the track. So we need to be thinking, preemptively thinking ahead about what the effect is going to be from the things that we're doing now. So obviously a key part of that is to get to know the biology, get to know what's actually happening there. And if you do a biological sample, you will get a test back that talks about all these different aspects of the biological food chain, or the Soil Food Web, if you like. You can get a lot of information too from the Soil Food Web Inc. website. So if you're interested in that, just type in Soil Food Web Inc. Australia, and you can go on there and download different bits of information. Will go into this in real detail. I'm just going to skip over it here because we don't obviously have a lot of time. So the first, soil organism group that we'd like to talk about is algae. Now, a lot of people forget the algae. They sort of start off with the bacteria and they don't sort of go to that previous step, which is the algae, which is there as a food for bacteria. and is also living on those soil water pockets around our soil pores. And as this gentleman in front here said before, we do need soil moisture because we do need to foster those algal populations in order to be able to feed the rest of our food chain. A lot of time I get asked questions about, well, we can't establish a biological farming system here because it's too dry. Well, that's just incorrect. There are different species that grow in low rainfall environments that don't grow in high rainfall environments, and the species will adapt over time. But we have to look at how can we get those endemic species that have evolved for that situation, how can we foster those to grow? And one of the problems I think with the inoculant idea is that if we're brewing an inoculant that's being brewed in a laboratory, for instance, that doesn't have that local adaptation, then we're not going to get that. Whereas if we're doing compost and compost tea and we can add in local materials and local compost or local leaf litter out of our natural systems or out of our natural pastures, then we're going to, I think, be a lot further ahead of the 8 ball there. So if we look above algae, we start to talk about bacteria. Now, bacteria are a major storage force in terms of nutrients. They store nutrients in their cell walls, and they also convert nutrients over. So we're getting a lot of nutrient cycling happening there. And then above that, we're talking about protozoa. Protozoa, a couple of different types of protozoa, which I won't go into here because it can be a little bit, technical. But basically, the protozoa are consuming the bacteria at large rates. They're turning over those nutrients that are stored in the bacterial cell walls. They're cycling those nutrients. A lot of the soil testing that I do with my clients, and especially biological samples that I get back, the protozoa is non-existent. There's no protozoa there. It might have a little bit of bacteria, but there's no protozoa. So what happens when you've got all these nutrients stored in the bacteria, but they're not getting released because nothing's eating them, nothing's exchanging the energy with them, and putting out the nutrients that they've been storing in the cell walls, basically. So there's a huge potential there to unlock that with protozoa. The next scary word on there is nematodes. I guess it's scary if you've had problems with root feeding nematodes before, or stem or bulb feeding nematodes. And again, when I look at my samples that I get back from my clients, a lot of the time there's a predominance of the bad nematodes, and there's hardly any predatory nematodes, and there's hardly any fungal feeding nematodes or bacterial feeding nematodes. Now, when you have a look at a nematode under the microscope, particularly a bacterial feeding nematode, you can have a look in its digestive tract where it's eaten all this bacteria. Now, all that is the potential for nutrients to be released out of the bacterial cell walls again. So, similar to protozoa, we've got the nematodes, we don't have the nematodes there, we need to bring them in, we need to get them in there in the right amounts and the right species in order to do what we need them to do. And we want to limit the amount of fungal and root feeding nematodes. We still want to have some fungal feeding nematodes because they're going to release nutrients, but we don't want to have an influx of fungal feeding nematodes and an absence of anything else. So fungi, precious fungi, we obviously have heard about mycorrhizal fungi before in terms of living on living in the root zone rather. And as a host, they require a plant root system in order to be able to feed them the sugary exudates, in order to be able to track them to the root zone, in order for them to be able to work to produce nutrients and be able to feed it into the plant. If we could just get the fungal levels up on our farms by an extra 100%, which sounds huge, but if we could just, if we're starting off at zero or just a very small amount, if we can just start to increase that over time and build that up to a point where it's there and we just have to manage it, then I think that's going to be real potential for the future. I think that's a question. No? Oh, sorry.
What sort of things you're doing, Elise, in this area or wherever you're working to get all those set that?
Yeah, what's some of the things that I'm doing? So compost tea, compost extractions, composting, inoculants obviously going out with a seed, and I will talk about that on one of the other slides, and I just noticed that the time is getting away from us, so I'll keep going and I'll mention that at the end, and we'll go over that a bit further. So a lot of the time when we're thinking about the soil biological organisms, we forget to talk about the microfauna and the macrofauna. Micro obviously being small, macro being larger. Most of the time when we talk about biological farming, people come and say, oh, it's all about worms, or I've got a few worms in my garden. Well, that's really nice, and you can see those worms, that's good. But there's other things other than worms that cycle nutrients. In the microfauna aspect, we're talking about mites and gnats and springtails and very tiny little things that we can't necessarily see with our naked eye that are sitting there that are cycling over those nutrients from further down the food chain from protozoa and fungi and things like that. And obviously the macrofauna, everyone's seen spiders and ants and worms and millipedes and things like that that are creating the pathways for the biology to live in. A lot of the time we're forgetting that those soil pores that the biology have to live in, the macrofauna are designed to actually create those. Worms are designed to go along, burrow down, take particles of organic matter, actually deposit them further down the profile so that there is actually that tube there and an airspace and water pocket for moisture to infiltrate and the biology to then come along, colonise and live in there and to start that cycle going. And I just thought I'd put in for a comparison some different photos from underneath the microscope of dead soil versus live soil. And you have to forgive me because there's only so much you can photograph down the eyepiece of a microscope, so it's not very wide-ranging. And obviously with multiple shots I probably could have put together a bit bigger picture, but we could be here all day if I was doing that. This slide here just demonstrates basically soil particles, mineral particles. There's really not anything really alive there. There might be some bacteria if you focus in and focus out, but there's not a whole lot of activity happening there. And I can't remember where I got that soil from. I think it was just out of the driveway. So not much activity happening there, probably very compacted, probably quite sandy. It's probably had applications of gravel and things over time to actually limit growth there. And obviously you don't see many things growing in your driveway because it's not a very hospitable place for plants, and therefore it's not hospitable for microbes. If we were to compare that to a more interesting soil, we can have a look at this picture here and see that we've got this browny tinge in the top left-hand corner, which is an indication of humic acids that are leaching out of the soil, so humus sites that are become soluble and feeding those bacteria there. We've got some fungal spores. We've got a little piece of plant root up in the top corner that's being decayed, and you can't actually see it, but there's some very small fungi. It's probably not very visible on here, but some very small fungi up there actually digesting that root material. So we started to get a bigger picture. In the same slide, I couldn't put all the photos on there, but we had amoeba, we had mites, we had nematodes, we had large networks of fungal hyphae, or the fungal vegetative phases up there. So we started to put together this picture of, wow, this looks really interesting. There's lots of things happening there versus there's nothing happening here. And when you compare the two shots above ground in terms of what's actually growing there, you've got a diverse habitat, fungal actually fruiting bodies on top of the soil. So when you see mushrooms and things, they're just the fruiting body coming out from the vegetative phase that's underneath the ground. And just lastly, just to finish up so I don't take up too much more time, we need to try and get that biology out there. So we're hoping that in the future that we don't have to keep continuing to do sprays of compost tea and to do compost, because we're hoping that we can get there to a sustainable cycle where it is actually then we're just maintaining that and monitoring it over time, preventing any negative effects happening to the biology. But for the time being, while we're working from behind the 8 ball and we've actually minimised our diversity, we've got no fungi there and we've got very little of the other soil organisms, we're trying to do what's called an extraction process, basically, where you take some material that is it's been analysed under the microscope that has all the goodies in it, and you extract that in a water process, so usually through an air blower, and actually agitate that water around the material, usually in a compost tea bag, and extract those organisms off there into the water. So you're basically making an infusion, taking the organisms off, putting them into water and spraying them out that way. Quite an efficient process because you don't actually have to brew it. You're just taking the organisms off and putting them into the water and getting them out there. Brewing's a little bit different. So you go through the extraction process, you start to extract that, take the organisms off the organic material, get them into the water, and then you start to feed them and brew them with increased oxygen amounts. And obviously the food provides them, a source of food to grow, and you get some bacterial brewing and protozoal brewing that way, and then you'll have a lot more concentrated, compost tea to put out there. And obviously to do that, you also need some compost to start off with to put in the tea bag. So you don't actually have to have large windrows of compost. People think, oh God, we have to make, like 1 million tonnes of compost to fix my farm. Well, you can actually start making just really small amounts that you can make in your own home backyard. You might even have already attempted to make compost before, and use those small amounts to brew up with compost tea. So compost tea is a lot more cost-effective way of getting it out there, because for most people, they don't have the compost there already on their farm. They have to buy it in, and at about $110 probably minimum per tonne, you're looking at quite an expensive process there to actually cover your whole farm in compost. So if you can make it yourself in small amounts and extract it out or brew it out and get it back out there, then it's going to be a lot better process for the back pocket. And over time, it will have the same effect anyway. You might want to put out, if you've got a really average paddock, you might want to put out a little, tiny little application of compost and then follow up with compost teas. And the other aspect of it is inoculating your seed. Obviously, you can buy commercial inoculants, but you can also do it yourself inoculant. Basically, make up some compost tea and do a seed treatment. It's a lot cheaper way of getting it out there. You don't have to be going out there with multiple applications over the paddock spraying it out. Obviously, that might be a quicker way of getting it out by spraying it out there, but if you've only got one option to do it, well, get it out on the seed where it's going to have that most effect. As soon as that seed germinates, those roots start to grow out, it's going to be in contact with that biology and it's going to start to grow, and the root will provide a host for fungi and the bacteria and things will start to happen. You've got the food out there and you're managing it properly, then it's going to work. And you've probably all seen, photos of nodulation and rhizobia, etc. But I just found this one the other day at the farm on a biological paddock that we've been doing, and you probably can't see from the back there, but if you can get a hold of this photo later, I can send it off to you. But there's just so much nodulation happening there, and they're beautiful and pink and really active. And it's only a small clover plant, but you can just see that as it's going to get bigger, it's going to start to grow and develop. And that's what we really need to be focusing about is all, getting like really down to that scale where we're not seeing it, but we need to be aware of what's happening.